Good afternoon, everybody, and thank you for joining us in this talk. The main talk will be held here by Ervin today. My name is Julia, and I'm going to represent the Center for Translational Genomics today. Annabella asked me to inform you that there are more interesting seminars upcoming, so please feel free to register if you find any of these topics of interest. And check out our LinkedIn, right? SciLifeLab. Yeah. One second. No. I think, what do you? Okay, thank you so much. As said, I'm part of the Center for Translational Genomics Lund. We are also called Clinical Genomics Lund, and we are part of SciLifeLab. Before we move to Ervin's talk, I would like to briefly introduce our work and mission. CDG is an open next-generation sequencing core at the medical faculty here at Lund University. We are providing advanced genomic services and expertise to researchers in academia, in industry, and healthcare. While our primary focus is local, we serve researchers throughout Sweden and even have some customers in Denmark. You can also find us under the name, as I mentioned before, Clinical Genomics Lund, as we are part of the Clinical Genomics technology platform within SciLifeLab. For those unfamiliar, SciLifeLab is the national research infrastructure that unites all major universities in Sweden. It is organized in 10 technology platforms and 40 research units. And it is providing access to advanced technologies, state-of-the-art instrumentation, and expert know-how. And one of these platforms is Clinical Genomics. The platform operates through 7 nodes, each located closely to a university hospital. So we have Umeå in the north, Lund in the south, and then Uppsala, Stockholm, Örebro, Linköping, and Gothenburg. The platform collaborates closely with healthcare providers and is a key driver of genomic medicine Sweden, pioneering innovative approaches to personalized medicine. The different nodes focus on different technologies, and here in Lund, we are leading the single-cell and spatial applications. So what is the mission behind clinical genomics? We are here to bridge the gap between basic research and clinical practice to enable genomic discoveries to be integrated into healthcare. And by genomic, I mean we are focusing on high throughput genomic-based technologies, like you can see here, sequencing, short and long read, single cell and spatial omics, liquid biopsies and ultrasensitive variant detection, multi-omics, metagenomics, informatics, AI, and data sharing. So how does this work in practice here at CTG? We participate in GMS work packages, such as focused trials within Testbed Sweden, or we take part in the National Genomic Pandemic Preparedness Portfolio G3P initiative. And once methods are validated for clinical use, there are transitions to units like the Center for Molecular Diagnostics within Region Skåne, ensuring a seamless transition to healthcare providers. So here at Lund University, we offer a broad range of services from DNA and RNA sequencing to advanced applications like single-cell and spatial genomics. In 2024 alone, we provided over 40 consultations, conducted over 200 customer projects, and processed over 4,000 samples. Our work from previous years contributed to 24 publications in 2024, and one will be highlighted now from Ervin. And therefore, now it's my pleasure to introduce Ervin to you. He's a PhD student in his last year before the defense in summer with the Pereira group. And you are an expert on reprogramming antigen-presenting cells. And you will discuss now your groundbreaking work on reprogramming solid tumors into dendritic cells for cancer immunity. So thank you for being here today.
Thanks so much for these very nice words, very complimenting words, and also for the invitation to present to you today. So what we have been working on, so I'm a PhD student at the end of this building. So if you have questions, if you want to discuss also afterwards, you know where to find me in A12 in Philippe's group. So our aim and our study that we recently brought out was to develop a new cancer immunotherapy modality by reprogramming cancer cells into dendritic cells that can then elicit an immune response against the tumor itself. But of course, before I go now into the project, I don't know your backgrounds, if you're familiar with cancer immunotherapy, how much you know, but up until now, I think many have heard about the most successful immunotherapy modality, which are immune checkpoints. But we can stay here just quick and acknowledge how much work there was put in the field during the last, I would say, even 100 years, because immunotherapy in cancer wasn't really successful for a long time. And just with the emergence and understanding of how the immune system interacts with cancer, we got closer to actually treating cancer with immune therapy modalities. And now that we know it's possible, and particularly with checkpoint inhibitors, we found out that it's possible. There's now new modalities emerging in the field. Some of them are highlighted here, like bispecific antibodies are one of the most recent ones that were added to the landscape of immunotherapies, adoptive T cells, cancer vaccines. But so far still, checkpoint inhibitors have been the most successful ones, tested in over 50 types of cancer, such as PD-1 and CTLA-4. And what's important to know is that for the first time, cancer immunotherapy has led to actual long-term survival in patients, because that was not possible with chemotherapy, radiotherapy. This was just extending a life. But now for the first time, physicians talk about cured patients. And we see that it is very variable across different cancer types. So this is graphics that shows the responder rate for anti-PD-1, which is so far one of the most successful checkpoint inhibitors and general immunotherapy modalities. And we see that it's an agnostic therapy, so it can be applied to different types of cancer. But as you can see, only in melanoma, it has a really high percentage of responders. So not all of these are long-term responders. These are also including partial responders. And then we can see across other types of cancers, we have even less. So there's still a high need to develop better immunotherapy modalities. And there's still a lot of patients that do not respond at all to immunotherapy. And of course, researchers, physicians are trying to investigate what's the reason. Why do some patients respond? Why others not respond? And there's a lot of reasons. I just summarized a few reasons down here. And this is, even that to summarize, is a lack of an immune response against the tumor. So for example, lack of antigen presentation in the tumor that will elicit anti-tumor immunity, an immunosuppressive microenvironment, heterogeneity in the tumor, but the list goes on. So there's many reasons why there is lack of immune responses. And of course, if there's a lack of an immune response, taking off the brake of immunity with checkpoints, it will not help, because there's not, in the beginning, to begin with, a good immune response. But now, to come to the problem, who is the one initiating the immune response? And that has been mostly described by dendritic cells, particularly the dendritic cell type 1 is very critical in the initial immune response. So it does elicit many functions. So now here we have a summary of several functions. Recently, last year or 2 years ago, there was a paper by the Böttcher lab where they described dendritic cells type 1 being able to initiate those hubs with stem-like T cells that can then infiltrate the tumor tissue and act as effector T cells to initiate this cytotoxicity against tumor cells. then dendritic cells type 1 can also migrate to tumor-draining lymph nodes where they interact with naive T cells to initiate de novo T cell responses. Then resident dendritic cells that have been described as this immunostimulatory phenotype, so expressing high MHC molecules on the surface and also cytokines and chemokines, are recruiting effector T cells then to the tumor to inflame the tumor and elicit an immune response. And then one of the most recent publications, it's a preprint by Miriam Merad's paper. They found that dendritic cells type 1 can even initiate tertiary lymphoid structures and maintain them. And this is very important and as has been found as a biomarker for positive correlation with a response. So dendritic cell type 1 are very critical in initiating immune responses. However, we don't have really a way to generate dendritic cells. And there's also clinical data suggesting the more dendritic cells you have in the tumor, especially the type 1, the more T cells you have. It's across several types of cancer, the dendritic cell type 1 that is positively correlated with survival. And if you combine it with checkpoints, so a high DC signature in the tumor, combined with a checkpoint inhibitor treatment, you have better survival. Indicating again, if you have an immune response and you add checkpoints, they will survive better. So as I mentioned, there is no method so far to generate a pure population of CDC1s for therapy. And there is many approaches that have been tried, not many work. And in our lab, we are specialized on direct cell reprogramming. So we thought this could be a way to generate a cell type of interest that you need for treatment. And there has been other approaches already described by many other pioneers in the field, how you can reprogram a somatic cell type, such as a fibroblast, a skin cell, into other cell types that will then elicit the function they need to. And this is usually done by the overexpression of lineage-specific transcription factors. Taking this to the next step, you can even deliver these transcription factor combination cocktails to the tissue itself in an in vivo environment. So in the mouse, it was tested already, to reprogram tissue in the cell type you need. There's been examples such as pancreatic cells that have been reprogrammed into beta islet cells that can produce insulin. There's been fibroblasts in the heart after a heart attack reprogrammed into cardiomyocytes that can actually really bump and lead to improved heart function. So there's many examples. Many of those, most of those have not been really close to the clinic. This example is now reaching almost the clinic, but they have, because they have one big limitation. You need to develop a good delivery vector to deliver the transcription factors to the tissue. And you need to prove that the same transcription factor combination that works in vitro can also work in vivo because the environment is different. So our lab has identified, that was in 2018, we published that 3 transcription factors, PO1, RF8, and BAT3, can reprogram fibroblasts into dendritic cells type 1. So those are very similar to natural dendritic cells, induced DC1s. They do perform similar functions such as cross-presentation, MHC2 antigen presentation, cytokine secretion, chemokine secretion. It's a very good system. And we hypothesized that if we use the same transcription factors and deliver them by a lentiviral construct to cancer cells, we can reprogram a cancer cell as well into an antigen-presenting cell that will present its own tumor antigens to the immune system and elicit anti-tumor response. And that was published last year in 2023, actually 2 years ago now. And we showed that tumor cells using cell lines from humans and mice and also patient samples, when reprogrammed with this lentiviral construct, this polycistronic construct with PO1, RF8, and BAT3, they remodel the chromatin of a tumor cell. They acquire a CDC1 transcriptome. They upregulate the antigen presentation and processing machinery, and they increase their immunogenicity. And when injected then into tumors, elicit immunity. We see longer survival of mice, and this also combined with checkpoint inhibitors synergizes, which leads to even more responders. Here, the problem would be that what we've shown here is that we reprogram cancer cells in vitro, which would require a protocol to isolate tumor cells from a patient, reprogram them in vitro, sorting, re-injecting into patients, which is difficult. And that's a long-term protocol. Complex ex vivo manipulation is usually something you want to avoid if you want to go to the clinics. That's why we hypothesized now, can we transform this into an in vivo reprogramming approach? Can we develop a vector that can deliver the transcription factors directly to the tumor and then reprogram inside of the tumor, the cancer cells into dendritic cells that will do the job? And that's why we worked on this study where we first wanted to check that we can reprogram cancer cells in vivo. So we used mouse cancer cells, whether they will elicit anti-tumor immunity, which immune mechanisms they are eliciting. Then next, to give it a translational point of view, can we reprogram human cancer cells? And for this, we used spheroid models. What is the role of immunosuppression? Usually, tumors are immunosuppressive. Will immunosuppression have an effect on cancer cells acquiring an immunogenic phenotype? And then lastly, can we develop a vector to efficiently deliver the transcription factors to tumors? So for this study, we used mostly for the mouse part of the study 4 melanoma cell lines that are very different in their properties. We have this B16 line that is very low immunogenic, hard to treat, grows very aggressively. This BRAF melanoma cell line that in bud-free knockout animals, so mice that don't have endogenous CDC1s, also grows very aggressively. YAM 1.7, medium immunogenicity, doesn't respond to checkpoint inhibitors. So, and B2905 that usually responds to checkpoint inhibitors. So we have a good range of cell lines to test. So what we started with was using the lentiviral construct, encoding the 3 transcription factors. In addition, we had a construct that is empty, just encoding GFP as a control. And then in addition, we used macrophage reprogramming vector, which we cloned, using macrophage transcription factors PO1 and CBP alpha that was previously published to reprogram fibroblasts and also cancer cells into macrophage-like cells, another antigen-presenting cell type. And by this, we can control whether really the CDC1, the unique CDC1 properties are the ones that are important for anti-tumor immunity. So we transduced cells in vitro, and then right after transduction, implanted them in the animals subcutaneously to establish tumors and allow the cells that were transduced, delivered with the factors, reprogram in vivo within the tumors, because reprogramming takes around 9 days. And then we did this either as a monotherapy, inject isotype antibodies, or combined it with checkpoint inhibitors to see what will happen. And very interestingly, in this aggressive B16 model, we saw in the monotherapy setting already better survival with the 3 transcription factors only. And we saw some that completely responded. So that was for the first time that we saw that in vivo reprogramming can lead to complete tumor regression. And when combined with checkpoint inhibitors, anti-PD-1 and CTLA-4, interestingly, all animals survived up to 100 days, 80 days without any tumors. That was quite striking for us to see as a first experiment. We saw also on the animals where we implanted the melanoma tumors, these white spots as a response against melanoma antigens. So this is usually a sign of, it's called vitiligo, a sign of a cytotoxic melanoma antigen response. So we tested this in the other models, and we saw a good delay with B16 again. Here we reduced the ratio of transduced cells to untransduced cells to make it a bit more challenging. And here we see that for this model, usually we would need checkpoint inhibitors to induce complete regression and not only partial responses. While in the YAM 1.7 and B2905, already as a monotherapy, we have complete responders. Especially here, it was interesting that all animals that were treated responded. We also observed in the blood, when combined with checkpoint inhibitors, which are the high bars here, a high percentage of T cells that are responding to tumor antigens. So these T cells are from the blood, they are circulating, indicating they are systemically around in the blood. That was a sign of systemic immunity, which we later wanted functionally to prove as well. In the bud-free knockout animals, which again, just to remind you, don't have endogenous CDC1s, we implanted this BRAF melanoma model and used in vivo reprogramming and saw as monotreatment or with anti-PD-1 even stronger the response, indicating that we don't need endogenous CDC1s for that. It's really dependent on the in vivo CDC1 reprogramming. As in many other vaccine studies, it has been shown that when you vaccinate animals with dendritic cells that were generated ex vivo, usually they just transfer antigens to endogenous CDC1s. That will lead to a response, which is not in this case. So we re-challenged the animals after 100 days so this only the survivor animals. And these survivor animals established really strong immune memory. So most of the animals could survive without any tumor outgrowth, indicating that they were resistant to relapse. We also rechallenged bat-free knockout animals, so the CDC1 deficient animals. So once immune memory is established, you don't need endogenous CDC1s anymore. It's very likely T-cell dependent. So coming back to the systemic immunity, what I mentioned before, what we did was this very sophisticated experiment with 3 tumors per mouse. So we implanted on the right side tumors that contain in vivo reprogrammed cells. On the left side, tumors that have no in vivo reprogramming, so just tumor cells. And then the third tumor was a different type of cancer. It's a lung cancer that is not sharing antigens to see whether the immune response is also tumor specific, very likely tumor antigen specific. And what we saw is they're treated. So the right tumor, you can see here the red line is the tumor growth. And on the left side, the non-treated, both are controlled very well by in vivo reprogramming, indicating that the response is systemic, controlling also the other side. While the lung tumor here is not controlled. That explains very well that we have a systemic immunity, which is tumor specific. And you can see exemplative images here from the mice with the small tumors on each side, while the controls are growing very fast. So then we looked into the tumors. How do they look? What is happening inside of the tumors? We performed this imaging by confocal and saw that in vivo reprogramming on this side induced these huge structures within the tumors that are full with CD45 positive cells. So we hypothesized those could be tertiary lymphoid structures, another biomarker of a good response. And the first time that we could actually connect CDC1s with tertiary lymphoid structures. And we confirmed that indeed in these structures we have CD19 positive cells, B cells, CD4 positive T cells, CD8 T cells, and those podoplanin positive stromal cells that are usually characterized to be around and inside of those TLS, indicating that indeed these are TLS structures. So we characterize further by flow cytometry how the immunophenotype of those tumors changes, and we see this big influx of CD45 positive cells, immune cells, which is composed of B cells, NK cells, CD4 T cells, and we did not see a difference in the CD8 T cell infiltration, which was a bit surprising to us because we expected a lot of CD8 T cells to infiltrate those tumors. But when we characterized the T cells more in detail in their phenotype, we saw that it is less, so these T cells are less exhausted, so probably also more functional, the ones that are there. And we have less regulatory CD8 and Tregs. Also, we have more stem-like T cells, which goes along with literature that CDC1s usually are connected with stem-like T cells in this inducing a response, a persistent response. Then we wanted to know how long are our reprogrammed cells inside of tumors. And we isolated the tumors at different time points, looked at cells that are GFP positive in CD45.1 animals, and found they are around 9 days in the tumor, and afterwards they fall down. So we don't see them anymore, and they don't migrate to lymph nodes. So they do acquire this resident phenotype, not a migratory phenotype, which we also confirmed by CCR7 expression, a marker for migration. And we used our bulk sequencing data set and found that indeed they express genes that are more associated with a resident signature rather than a migratory signature, which is over here. So then we depleted T cells and NK cells to find out which are the critical T cell, these effector T cells that are leading to the immune response. And to our surprise, we found that CD4 T cells are critical for immunity, as when we depleted them, tumors grew out very fast. And it was not the CD8 T cells, although we saw a bit of an outgrowth of tumors a bit in later days. So indicating maybe there's more of a memory response there. So then we performed TCR, single cell TCR sequencing, analyzed by our computational postdoc Ilya, and it was also with the big support of CTG. So we sent our samples here to be sequenced. We used blood T cells, tumor draining lymph node T cells on day 21 after implantation of tumors. And when we look at the CD8 T cell compartment, we see a bigger proportion of effector T cells in the tumor, especially when we combine it with checkpoint inhibitors. We see effector memory T cells and more stem-like CD8 T cells when combined with checkpoints, and less exhausted T cells. So when we put trajectories through those populations, we can see that the CD8 T cells are preferentially differentiating towards a memory phenotype after being an effector phenotype rather than towards exhaustion, which usually happens in non-treated tumors. We did the same for CD4 T cells, and we found, very interestingly, a cluster that we called cytotoxic CD4 T cells. So they have cytotoxic granules, they have cytotoxic markers that were kind of described in the last few years to pop up more and more. So there is more and more publications really indicating that CD4 T cells have also cytotoxic roles in the tumor. And this is what we found as well. Also, combined with our data that CD4 T cells are necessary for anti-tumor immunity, this made sense that we found this big cluster of cytotoxic CD4 T cells. And as well, when we looked at the differentiation, we saw less differentiating towards this exhausted population, which is up here, and more towards a cytotoxic population. So then we looked at the clones inside of tumors, and indeed we did not see really clonal expansion in the CD8 T cell compartment, indicating that they do play less of a role in these tumors. And we found in the tumors more clonal expansion in the CD4 T cell compartment, particularly in the cytotoxic cluster and in the Th1, in the T helper 1 cluster, which are described to be pro-inflammatory as well. So this was the part about the mice. Now, the human part, we wanted to know, can human cells as well reprogram in vivo? So we implanted transduced cells using glioblastoma, human glioblastoma cell lines, and melanoma cell lines in NSG mice, so immunodeficient mice, formed a tumor, isolated the tumor after a few days of reprogramming, and checked the expression of the reprogramming markers CD45 and HLA-DR. And we found that indeed they do reprogram in vivo. And interestingly, when we compare it to in vitro reprogramming, at similar efficiencies. So we don't see really big differences between in vitro and in vivo reprogramming on the efficiency level. But what we did see was a difference in the acquisition of an actual CDC1 phenotype, such as CDC1, including CDC1 markers XCR1, which is a very specific marker to CDC1s, and CLEC9A, which is acquired more in vivo than in vitro, indicating there's something in the environment that helps them become more like a CDC1. So then Malavika and Olga, former postdoc in our lab and the research assistant in our lab, formed spheroids and tested reprogramming in spheroids in 3D compartments across a lot of different cancer cell lines. And we found that all of them reprogrammed to a certain degree also in 3D, similarly to in vitro, in a 2D layer, which is important thinking about translational further on when we want to reprogram different types of tumors as an agnostic treatment. We performed single cell sequencing, and there is another interesting observation that goes along with the acquisition of the CDC1 phenotype more in vivo. We found that in 3D, when we reprogram cancer cells, they acquire a transcriptomic signature that is closer to actual CDC1s earlier in reprogramming. So already on day 3, if you look at the 3D tissue, they start affiliating more to DC1s than when we compare it to in vitro, which then happens later on as well in vitro, and even stronger in 3D. So I always say in vitro, but I mean 2D. And as well, our own in-house tumor APC signature, which we created beforehand to represent how much they actually become antigen-presenting cells. Then we performed gene set enrichment analysis and found that the cells that reprogrammed acquire also a mature immunogenic CDC1 phenotype. A program, not phenotype. Then the immunosuppression, also a very important topic. So we formed spheroids, so we could model immunosuppressive spheroids, added cancer-associated fibroblasts, myeloid-derived suppressor cells, or pericytes, all described as usually immunosuppressive cell types. Together with glioblastoma cells, added the transcription factors encoded by a lentiviral construct, and found that there is actually no impact on the reprogramming efficiency. In fact, we even see here a bit of a better reprogramming efficiency when we add myeloid-derived suppressor cells, indicating that reprogramming progresses a bit differently than what usually is known in immunotherapy. So we wanted to also test the function. Is the function impaired when we add immunosuppressive cells? And it was not, because if we add PBMCs from donors to the spheroids containing cancer cells that are reprogrammed or just enteric transduced, so the controls, we see no reduction when we add PBMCs that have not been pre-activated before. While we do see the reduction, so killing of the spheroid when we add them to spheroids containing reprogrammed cells. And we see high secretion of interferon gamma, TNF-alpha, granzyme B, which are usually cytotoxic cytokines. So the last part of the presentation is to develop a good vector, and this was done together with Asgard Therapeutics. This is a spin-off company from our lab, which aims to develop this treatment to bring it finally to the patient. And there was very good scientists, hardworking scientists that together with us were doing big screens of using the different vectors, lentiviral vectors, adenoviral vectors, or adeno-associated viral vectors in mouse and human cells, in 2D cultures, 3D cultures, in vivo, testing transduction, reprogramming function, but I will go quickly through this set. So there's more data behind that just to sum this part a bit up. Using patient-derived cancers, so we have melanoma cancers, lung, breast, head and neck in 2D and 3D. We see that lentiviral vectors, as well as adenoviral vectors, induce efficient reprogramming towards CDC1s, while adeno-associated vectors did not perform well. We did not see very good reprogramming efficiency. And then when we tested this in an in vivo situation, so we tested the vectors encoding only EGFP, so there are no transcription factors, it's only EGFP, to transduce B16 tumors in vivo by intratumoral injection. We saw that actually adenoviruses and AAVs perform the best. So they transduce cancer cells more efficiently than lentiviral vectors. So then combining the data from before, knowing that adenoviral vectors can induce efficient reprogramming, together with adenoviral vectors being good in transducing cells in vivo, we decided to follow up on adenovirus to continue working with this vector type and injected adenoviral vectors into this human sarcoma and melanoma tumors, established once more again in NSG mice, and found that indeed, when we inject them intratumorally and hit the cancer cells, they acquire a CDC1 phenotype. So it is possible to entirely reprogram in vivo, even by in vivo delivery. So then what was left, the final experiment, can we induce immunity by this treatment? So we chose again the most challenging model of the 4 models that we have been using, B16, injected 4 times adenoviral vectors encoding PIB on day 7, 9, 11, and 13. Combined this with checkpoint inhibitors. As I showed you before, B16 requires checkpoint inhibitors to induce complete tumor regression, and then followed up on the animals. We saw that 4 out of 8 animals completely regressed in tumor size, while you can see the other control animals that have also got checkpoint inhibitors do not respond. So again, this is a non-responsive model. They survived 100 days without any tumor that grew out again. So we re-challenged them, and all 4 of the 4 animals survived without any tumor relapse. So we again re-challenged them, but this time intravenously to imitate metastatic node formation, because if you inject them intravenously, they will go to the lung. And compare this to naive animals that were age-matched but did not receive the treatment before. And we can see the lungs of the survivor animals are beautifully white. We've only found in a few small metastatic nodes, while in the naive animals, I mean, it's very obvious that they are full with black metastatic melanomal nodes. So to conclude this study that came out, I think 5-6 months ago, we found that adenoviral vectors can deliver the transcription factors PO1, RF8, and BAT3 efficiently to tumors, induce reprogramming in vivo into CDC1-like cells that express CDC1 markers such as XCR1, CD40, CLEC9A, and high expression of HLA molecules that are required for antigen presentation. And this leads then to immunity by forming tertiary lymphoid structures, establishing CD8 and CD4 T cell responses, interestingly, more clonal CD4 T cell responses. And these T cells are exiting then to the circulatory system, protecting the mouse from metastasis. So optimally, during the next years, we will see this landscape a bit changing with a new immunotherapy modality being established, which is called direct reprogramming, including our CDC1 reprogramming approach. This is what Asgard Therapeutics in the Medical Village is now working very hard on. And hopefully in a few years, we can see the first patients benefiting from this new modality. This is something that the group will continue working on. But here at this point, I want to acknowledge them for the work they have already put in. I want to also acknowledge the team of Asgard Therapeutics. They are very hard working on this to really bring this to the patient. I want to acknowledge all our collaborators. There's a lot of collaboration, and I could not fit all our collaborators on this slide. I hope nobody will be angry at me, but for example, Lina Olsson, Susanne Strömbler from LBIG, the Lund Stem Cell Center for Imaging, generally the Lund Stem Cell Center for all the good input, A12, also the Center for Translational Genomics for providing us with helpful sequencing data, the University Cancer Center, and I want to really personally thank Felipe Pereira for giving me this exciting project to work on during my PhD and all the help from the group that made this possible. That's it from my side. I hope you have questions.
Yeah, Ervin, for this really nice research project, really inspiring. Does anybody here in the audience have questions?
Yes.
Annabella?
Sorry, so my question is super basic, but are you trying with, in the case, mice, male, females, both together, combined? Because I guess there is one, there are relationships between them, right, immunology, and yeah. So this is my question, if you are mixing them or just trying them independently.
It's a very good question, something that should be more considered, I think, in immunotherapy. So in some of our models, such as the bat-free knockout models, we have mixed male and female because we are breeding them ourselves. So we can have access to both. In the other models, we are usually using female mice because they are easier to house because they will not fight with each other. In the future, I think it would be good to test what's the difference if we test it only in female versus only in male. Is there going to be different responses? From the data that I've been discussing with others, I heard that in male mice, there's usually a stronger immune reaction and better response to immunotherapy than in females. But that is only what I heard, not something that we found in our data. But I think also extending this question to, for example, testing in old mice versus young mice is going to be also very interesting because usually cancer is something that develops more in the elderly population. Well, this is tested mostly in young mice. So how is it with that? Also. But yeah, very good question. Yeah.
How can you be sure that body is developing at the same time? You put the cells inside, but how can you be sure? Then because the response will be different, right? Tumor is developing in a different way, in different animals.
So usually the animals are inbred, so they should be genetically the same and behave the same. So usually if you establish a tumor, you should, in theory, I'm saying in theory, develop the same tumors with the same kinetics. This is not happening. This is something that a lot of researchers have worked on to try to understand what can influence the phenotype of a tumor. For example, a B16 tumor is usually described as very cold, very aggressive. And usually when we implant it, it grows very out fast in the control animals. And you can see within 15, 20 days, they are dead because the tumor is too big. So you have to sacrifice them. Still, maybe from 10 animals, one will have a more immunogenic B16 tumor that will grow slower because there is an ongoing immune response. And researchers don't really understand why. Is it the location? Is it the time during the day that you implant? There's a lot of variables here. But of course, when you do these experiments, you do it for all the animals at the same time in the same location. So you try to minimize those variables. So usually then you can just really compare between treatment and non-treatment. But it's a big question. Why do some animals behave a bit different than other animals? In our models, we did not see so big variation because usually, especially if the B16 tumor is so low immunogenic, it will always develop.
Thanks for a nice talk. I was wondering about the reprogramming with transcription factors. Can you talk to how important the ratios in between the three different factors is, and if you have large tolerances or if you really need to max like certain ratios?
It's a very good question, something that we've been also working on, actually. So reprogramming generally, it always depends on the ratio of the individual transcription factors. So if you have, for example, in our case, we need to have high PU1 to initiate the reprogramming process. And we have tested different polycistronic constructs with having PU1 in the first place, RF8 on the second, and BAT3 on the third is the one that works the best in our case. But we have also tested them the other way around or intermixed to see what actually brings the highest reprogramming efficiency. And there's, of course, other places that induce reprogramming, but at lower efficiencies, because only some of the transduced cells will have high enough PO1 levels, for example, if it's on the second or third place. So it does matter at which position, or if you deliver them not within a polycistronic construct, you can also do RNA or something else, then you need to make sure that you have a good ratio of the individual transcription factors, but it does matter.
A follow-up question then. Yeah. When you, for example, deliver lenti constructs, do you ever see silencing parts? For example, the PD-1 will be silenced, but not the others.
I think that's, in our case, it's a bit difficult to look at that. We did not design the experiments to look at that. Mm-hmm. I do see with time, if we just transduce with GFP, that cells do silence it. But on the other side, they can also just be outgrown by cells that were not transduced. So it can be both. So it has to be sophisticatedly designed an experiment to analyze that. But it has been described that lentiviral constructs do get silenced over time because they integrate and then you just silence the region as a immune mechanism from the cell intrinsically. Can happen. But also in our side, we are reprogramming towards dendritic cells, and they are not dividing anymore. So they are post-mitotic cells. And what we see, cancer cells, they grow very fast, a lot. And as soon as they get reprogrammed, they reduce the growth. So they get overgrown by the other cancer cells. So it's not the silencing, it's the growth of the other cancer cells that happens. But still, it's not the experiment to analyze that. Yeah. But I think it's a very good point. And we tested also different promoters, different constructs. And it seems with the SFFE construct, it's the most efficient to keep the cells reprogrammed. And as soon as they are completely reprogrammed, they stay, and then they die after some time.
When you try to transform the cells in the tumor, how did you inject your lentiviral vector? Is it directly injected into the tumor or is some other thing?
It's in the tumor directly, because the tumor as a closed system, it's quite practical to just go because the tumors are subcutaneously implanted, so we can just hold it and then inject into the tumor. Optimally, we would develop a therapy that can be even injected intravenously and then target the tumor. That would be something that I think big pharma would be very interested in. Mm-hmm. It's much easier, but
we can check that whether the transformation which is happening is localized where you have injected it, or
it's very localized. So lentivirus generally transduces very inefficiently in vivo, but with adenoviral vectors, AAVs, we have checked where it goes if we inject intratumorally. And I think mostly if it leaks out, and it doesn't really leak much out because it's so contained in the tumor, it leaks out and would go to the liver because that's the highest tropism that adenoviral vectors have. So if you would inject it intravenously, they would go all in the liver.
But if it is localized, you inject it into the tumor and then only few cells around the injection site are transformed, would it be effective enough to
initiate immune response? Yeah, we have actually an experiment. I cut it out, but there is an experiment that we did. How many cells do we need for a strong immune response? So what we did is we diluted them down. We transduced the cells around 80%, I think, and then we added more and more untransduced cancer cells to the mixture and implanted in vivo. So that we started with a transduction of 50%, but then we went all the way down to, I think, a few percent, which translates into reprogramming efficiency because not all of the transduced cells will reprogram. So we had around 2% of reprogrammed cells, I think down to 0.0 something percent, 0.01 if I remember correctly. And we already at 0.1% of the cells in that mixture that are reprogrammed, we have enough strong immune response to so that the mice survive longer. And then at 0.3%, I think we already have the first complete survivors. And at 2.2%, we have all the mice surviving. So it's very low numbers of dendritic cells that you need to initiate the immune response. And it kind of makes sense from biology because if you have one dendritic cell that will activate a T cell that will then proliferate and amplify the response, it's enough. So it kind of makes sense with biology.
One last question.
Yeah.
Have you tried any other method to transduce the cells or deliver your transcription factors so that you like electroporation?
Yeah, we have another project in the lab. Nate's ARC, he's a PhD student as well in our lab. He's developing RNA-based reprogramming. So he tested delivering RNA. I think it was with lipofectamine. He usually transfected the cells, and he sees as well reprogramming with RNA. It's just not as efficient yet, I think, as with lentiviral construct because the lentiviral integrates. And here you need to deliver the RNA constantly to have enough levels of RNA to activate the endogenous transcription factor expression. And as soon as you activate the endogenous, then it's a self-sustained system because then they will be expressed and they will then lead to the stabilizing of the cell fate. But what he found out is that there is different RNA systems like self-amplifying RNA, just mRNA, circular RNA, and he tested different systems and they all work a little bit differently. But we still need to work on that to find what's the best way with RNA to induce reprogramming. Also, there's another project with small molecules. Yes, microRNA, yeah. Also, so there's a lot of different delivery systems that can be used, also small molecules, which would be also great if we find a cocktail of small molecules that can activate the endogenous transcription factors, because the easy of producing small molecules is very easy, and the delivery, because they are cell permeable and they're non-immunogenic. So that would be great to have a cocktail of small molecules. So there's now a parallel project ongoing to not only focus on the adenovirus, but going away from that to other delivery approaches. Yeah. Mm-hmm. Mm-hmm. Yeah, so actually, can go back. Here. So this part here has been done with the GeoMix from NanoString, and this part with the confocal. So we did already perform the spatial transcriptomics analysis and taking RNA from different spots from the TLS, from the GFP population, from the pan-CK, so just the cancer cell population around. And we are right now analyzing that. So I just came from a meeting with Ilya, our postdoc, we started analyzing that. But we'll for sure have to continue because also the nanostring has its downsides and upsides. So you can analyze not single cell resolution, you analyze the whole region, but you segment the cells, so you take them segmented. But then there is also other better approaches to look more in detail. So I think each approach has its own advantages and disadvantages, but we started going also in that direction because there's a lot of questions, but one question is, for example, why are the cells around these TLSs inducing the TLS here and not here? What is the difference between the cells here and here? Are those cells better reprogrammed than those cells here? Are they higher expressing transcription factors that leads to better reprogramming? Or are they just by accident there? So is this a stochastic process? So there's a lot of questions. Also, what's the difference between the TLS here and here? Can we use this approach to induce synthetic TLSs agnostically across different tumor types and make them responsive to immunotherapy, which would be another great application of that approach, not using it as a therapy itself, but using it as a method to induce TLSs. and then analyze how our TLS is formed, because we don't really know. So there is a lot here to explore as well.
More questions? I
If-
just had one more.
Yeah.
I mean, we now had plenty of questions. But what are the next steps? I mean, it worked now in mice really nicely, in xenografts. So what is the
So I
plan
think
to get it into
the
clinical
clinics.
trial?
So that's a big approach from Asgard now. They are focusing a lot in bringing it to the clinics. We are, of course, helping, but I think they are now focusing a lot on the critical experiments that you need to show to the agencies to bring it to the patient, because now we have the proof of concept. So now we have the critical data to show, okay, it works, we have efficacy. We have some initial safety data already in this study showing that we don't have any autoimmune responses, which is a big topic here as well. Autoimmunity is a big problem in immunotherapy generally. But we need to take another step and go more detail in safety to see, okay, the adenovirus vector, is it contained in the tumor? Will it leak out? What if you hit a bloodstream? If you inject it and then it goes to the blood, what will happen? Can we have side effects from this treatment? So all of these are important points to consider, and you need to provide to the agencies that it's safe, that we can go to the human. Also the dose, how many times you need to inject the vector, how high the titer needs to be of the vector. What's the toxicity? Can we go down in the vector, and then do we need to combine it with checkpoints, or can we use it as a mono treatment? Which tumors are we going to target? So there's a lot of questions, because this is a tumor agnostic treatment. So the question is, of course, what is the best cancer to start with to target? Because usually you have to know your indication. Or can you just target a lot of different cancers in a basket trial, which is one of our ideas, to test it in a basket trial, different types of cancers. But then those patients are usually I would say the last resort patients where no other treatment is working currently. And of course, it's going to be a high bar to reach if you need to show efficacy in those patients. So there's a lot of these considerations. But I think just to sum up, it's safety, dose, tolerability. So these are the directions where this is going now.
So very small hurdles right now.
Yeah, I mean, this was already some hurdles, but I think, yeah, I wouldn't say small. There's still some hurdles there.
So then, if there's no questions anymore, then thank you so much for presenting today.
Thanks for having me.
Yes. Hope to hear you at another point with the next great project.
Yes. And if you have questions, I'm in A12. You can always reach out to me or just come by.